So after taking a look at the genetic toolkit that we share in common with bilaterians to develop things like our digestive system and nervous system, let's take a look at the origin of limbs and the common genetic basis for the development of limbs in those bilaterians that have them. So as my slide says here, and as your textbook says, limbs provide one of the best case studies for the way that a shared genetic toolkit has been used over and over again, over hundreds of millions of years, generating tremendous amounts of biodiversity. So, despite differences in growth and form, mouse legs, fly legs, or for that matter, just to generalize, tetrapod legs and arthropod legs develop in response to the exact same network of regulatory genes. So in terms of differences in growth, when an arthropod or an insect limb develops, all the parts of the limb develop at the same time. Different parts of a leg all develop at once. When it comes to a tetrapod limb, that's not the case. Different parts develop at different points in development. So that's pretty profound. And of course, differences in form means if you compare the structure of a leg of a fly or insect and the structure of the limb of a tetrapod like a mouse or a frog or a lizard, then once again there's tremendous difference in the growth in the form of the limb, and yet there's the same network of regulatory genes is responsible for the growth and development of limbs. There are specifically 7 orthologous genes that delineate all the different aspects of limb development, anterior to posterior, so head to tail, dorsal to ventral, back to belly, and proximal to distal, from close to the body to away from the body. And so these 7 orthologous genes are controlling the exact same patterns of development in all bilaterians. Pretty remarkable. So take a look at the figure here out of your textbook and compare it and see how the network of relationships of these genes is the same. Now, what you might also notice is that the names of these genes are different. So, for example, HH and SHH, those might seem to be different genes, but in fact they're not. They're homologous. But the reason why they have different names, just like the other genes in these networks, is because they were discovered at different times in different laboratories, and so they were independently named. So they are the same in terms of homology. They're the same genes, but many of them have been given different names for that reason. And that's the big takeaway message here on this slide, is exactly that despite differences in growth and form in these different limbs, the genetic network is exactly the same, 7 orthologous genes. So the structures are convergent, meaning that limbs in an arthropod evolved independently from limbs in a tetrapod or a vertebrate. If you want to go all the way back to fish with paired fins, and then those of course gave rise to limbs. So the structures are convergent, but the genes that control the development of these structures are homologous, going all the way back to at least the common ancestor of all bilaterians. So now, if we turn our attention to the development and evolution of feet, that is, tetrapod limbs, we know from the fossil record and comparative anatomy that fins came earlier than tetrapod limbs. But still, there are mysteries that were out there in terms of exactly what triggered or what allowed the development of tetrapod limbs from pre-existing fins. So again, using a genetic perspective, what we could see is that the same Hox genes that establish where fin rays and bones develop in fish and tetrapods, the exact same Hox genes are used for both fins and limbs. But there's differences in how the genes are expressed. And so again, it seems to suggest that mutations that affected regulation, regulatory genes, are what mostly explain the development of tetrapod limbs or feet from fish. In both fish and tetrapods, they begin embryonically expressing both Hox A11 and Hox D13 genes, so homologous genes. And bones begin to develop near the body, like the humerus, or the bones that are in the fins of fish near the body, those begin to develop. Those Hox genes turn off, and in fish, then there's, and by the way, the bones that develop from mesoderm. So remember, mesoderm is one of those germ layers that bilaterian animals have, in addition to ectoderm and endoderm. So these bones develop from mesoderm in both fish and in tetrapods. So in fish, then those Hox genes are turned off, and then ectodermal tissue in what's called the apical ectodermal ridge continues to develop, and that produces the fin rays. Now in tetrapods, that a period of growth from that ectodermal tissue is relatively brief, and then the Hox genes, or at least the Hox D13 gene, is turned back on. So regulation-wise, something happened, a mutation happened to turn that gene back on a second time. And as a result of Hox D13 turning on a second time, that results in the development of wrist bones and digits. So when you have the ending comparison, like in the figure above, of a fin and a limb, it's the homology of those genes is there, but differences in regulation is what determines the development of a fin versus a foot. So let's look at the development of fins and feet just a little more carefully and take a look at a manipulative experiment that shows how this Hox D13 gene may have indeed been responsible for the development of limb bones in tetrapods. So, of course, looking at the fossil record, we know that fins first appear in vertebrates well over 400 million years ago. So the genetics for fins goes back very far. And of course, the phylogeny in chapter 4 that was presented and shows the relationships of different vertebrates is based on morphology. Very traditional approach using morphology. So as we just touched on the last slide, fish fins and tetrapod limbs share some similarities in early development. They basically start off the same way. But then fish, of course, form bony rays, and tetrapods form long bones in their arms and in their legs. So this leads to a hypothesis that there was a shift in the regions dedicated to different tissues in the developing bud. So once again, it's a regulatory hypothesis. There was a shift in how genes are being regulated, and that shift focused on different tissues developing in the limb bud. So back to this HOXD13 gene that seems to be really important. It produces proteins in the outer rim of a fin when it's just beginning to develop, and then it switches off. So that HOXD13 switches off, and then that apical ectodermal ridge takes over and produces the rays. Whereas in tetrapods, HOXD13 switches on twice. Switches on early in development to produce early bones like the humerus. Then it switches off. And then it switches back on in tetrapods and forms the long bones instead of fin rays. So as an experimental manipulation, biologists added a hormone to zebrafish embryos, and this hormone caused the zebrafish fin to express HOXD13. So intentionally using this hormone, knowing that it would potentially turn on HOXD13, has control over it. And sure enough, when this manipulative experiment was done, the zebrafish embryos developed cartilage in the same area, in the same region where a tetrapod develops its wrist and its digits. So what this suggests then is that the genes were in place, the homology was there, and regulation was modified. Tweaking those regulatory genes was able to switch from making a fin with rays in it to a limb with long bones adapted to mobility and locomotion on land. So I want to share one other example of how changes in regulatory genes can fine-tune certain structures and lead to, in this case, and in any case for that matter, helps explain an adaptive radiation. The origin of many species, in a relatively short period of time, evolving and filling different niches, doing different things. So the Galapagos finches are, of course, an excellent example of a local, regional adaptive radiation that occurred not that long ago. And so the primary idea here at the top of your slide, altering the timing, the location, and the intensity of expression of genes in our genetic toolkit can generate different shapes, different adaptations. So it doesn't necessarily take new genes. It just takes tweaking those existing genes and regulating what's going on. And again, I want to emphasize this point strongly, that a few decades ago, again, I make reference to like when I was a student, the dogma was that new genes, different genes, were responsible for these kinds of changes. And it's really a revelation and makes things a lot more simple with the discovery that it doesn't require new genes, but just regulating existing genes. I mean, this is really a profound and important point that helps us to understand potentially the evolution of all kinds of different lineages of life. Just regulating and tweaking existing genes produces amazing diversity. So here in our example, there's 18 species of Galapagos finches. They've evolved different shapes of beaks, and they've done this in under 1 million years. It's pretty quick, and they're adapted fundamentally to different kinds of foods, seeds, fruits, insects, etc. And what's being represented here is that two genes that are responsible for the development of beak shapes, bump 4 and calmodulin. And so what I've summarized here is the expression of different amounts of proteins from these genes is what gives rise to these differently shaped beaks. So just little tweaks in regulation. So for example, if there's low amounts of the bump 4 protein, then you get a narrow beak. If you have high amounts of the bump 4 protein, then you get a wide, deep beak. Okay. Calmodulin. If you have low amounts of calmodulin, then you have a short beak. If you have high amounts of calmodulin, you get a long beak. So by changing the levels of these master regulators, how much protein is being produced from each of these, it helps, once again, it helps to explain adaptive radiations, the rapid diversification of species, relatively short period of time, into a variety of open niches. So we can imagine that these tweaks of regulatory genes are responsible for all kinds of adaptive radiation. So I'm sure there's lots of research that will continue to focus on different groups and identify the genes that are responsible for different adaptive radiations. So next, let's take a look, pardon the pun, at eyes, evolving eyes, and the origin of eyes. Because again, it's been known for a long time, morphologically, anatomically, that many different kinds of animals have eyes. And these kinds of eyes are so different that it's apparent, it seems, that eyes evolved independently because there's such dramatic differences in structure of eyes. And they've popped up in different lineages. So the convergence of eyes seemed pretty apparent. But the eye is a very complex thing. There's a lot going on there anatomically with the physics to give rise to our perceptions of images, and very clear images, crisp images, to be able to see so well. So the eye was one of those adaptations that was pretty daunting to Charles Darwin to explain because of its complexity. And yeah, he's quoted as saying, "The eye to this day gives me a cold shudder," because of his inability to adequately explain the origin of eyes in a gradual kind of way. And don't forget William Paley, back in chapter 2. It's part of that natural theology concept, who in the early 1800s argued that complex structures such as the eye could only originate by an intelligent designer, with the argument being that there's so much complexity to the eye, and everything needs to fit together appropriately for an eye to work. So how possibly could something as complex as an eye originate by gradual step-by-step evolution? That was Paley's argument. So let's take a look at this. So eyes are completely absent in sponges, which are basal early animals, and placozoans, which are flat little animals. You wouldn't even hardly think of them as animals if you saw one. There's no differentiation of tissues, no organs. Almost looks like a multicellular amoeba crawling around. They don't have eyes. But eyes are present in eumetazoans, cnidarians, and bilaterians. And you can see that on the cladogram that's placed on the right side of the slide. No eyes in sponges, no eyes in placozoans, but eyes appear in various lineages of eumetazoans. So, but just the fact that there's such a huge diversity of eyes, as I've already mentioned here, suggests that they evolved multiple times independently. So let's take a look. Vertebrates have what are called C opsins on the surface of their photoreceptors, of our photoreceptors, and protostomes have a different photoreceptor, our opsins. And each of these opsin proteins has a different protein pathway to the brain. So these differences alone suggest a separate, different evolutionary origin. However, like with other things we've looked at here, recent research shows that both opsins, R opsins and C opsins, are found in vertebrates and invertebrates. They're found in both. So when did opsins originate? If they're found in both, then that implies that they didn't evolve independently. There's just been some tweaking in each lineage. And so when did opsins originate? Goes kind of far back in evolutionary history, perhaps. Let's take a look at that in the next slide. So it turns out, as you'll see here, that opsins have their origins in G protein coupled receptors that are found in eukaryotes, so GPCRs for short. And G protein coupled receptors relay messages into cells, at least in animals, after binding to the extracellular matrix. So that's what they do. Opsins, as it turns out, we can see through our genetics lens, originated through gene duplication. So good old gene duplication. And they're most closely related to the G protein coupled receptors that serve as a receptor for melatonin. So you can see, looking at the figure, at the cladogram, that goes way back. Melatonin is color coded in gray. And so you can see, going back basically to the common ancestor of all animals except sponges, so including those placozoans, about 1 billion years ago, an ancestral gene for G protein coupled receptor duplicated, and one of those copies evolved into a receptor for the melatonin hormone, and the other diversified into opsins. So there we go. There's where our opsins originated. So now opsins are found in eumetazoans and placozoans, but placozoans don't have eyes. So it's interesting. Then after that event, gene duplications produced the C and the R opsins in eumetazoans. And again, if that, it bears out looking at this kind of complex cladogram that's showing the pattern of origins of these different photoreceptors. And so we can figure that opsins originally, when they evolved, probably clumped together and congregated together into eye spots to sense light and dark. And that would give rise to the first early eyes in various lineages. That would be the starting point. In many different lineages for the origin of eyes is these opsins, these photoreceptors that evolved way back before the common ancestor of placozoans to be able to have a platform for making eyes. So still on the subject of eyes, the origin and evolution of eyes, let's focus on lenses. So there's a protein that's found in the lenses of eyes, and this protein is called crystallin. And it's a remarkable protein. It's a very tough, durable protein, and it's essentially transparent. Light can pass through it. And so as a component of lenses, as a primary component of lenses, crystallin bends light, and by bending the light focuses images, focuses the light on parts of the eye to produce images. So that's how our images are able to be formed, because of the crystallins and how they work, the physics of crystallins in a lens of an eye. And so what we've discovered is that gene recruitment has resulted in the evolution of crystallins several times. And the gene recruitment has happened from a pre-existing gene that produced a type of protein called a shock protein, heat shock protein. These are proteins that are very stable and would hence be a great platform for making a highly stable protein for a lens. And so it's evolved several times through gene recruitment in cnidarians, because, for example, different lineages of jellyfish have eyes, vertebrates, arthropods, of course, mollusks, squids, octopuses. But to focus on vertebrates, so that's just saying right there, the lens of different eyes can be traced back to gene recruitment using the heat shock proteins and transforming them into crystalline proteins, which then could be used in lenses. But if we focus just on vertebrates, on the vertebrate eye, the alpha crystalline, which is what we have alpha crystallins, are homologous, again, to pre-existing shock proteins, very stable proteins. And so the figure on the right-hand side of our slide kind of shows the evolution of vertebrate eyes. So in early chordates, we essentially have eye spots. In fact, that little lancelet that you probably looked at in Bio 182 has little kind of eye spots along its back near its dorsal hollow nerve cord. And in hagfish, hagfish have a cup-like eye that's filled with crystallins. And so it gives a fuzzy image. They can see images, but it's not very clear. In lampreys, which some biologists argue are more closely related to us gnathostomes, other evidence suggests hagfish and lampreys are sister taxa. But lampreys have a lens in their eyes. So do we. So if lampreys are more closely related to us gnathostomes, then we could say the lens is probably homologous for lampreys and us. If lampreys, in fact, are more closely related to hagfish, then maybe the lens evolved 2 times in lampreys and us. That's a side issue. So we get a lens in lampreys, and then finally in gnathostomes, sharks, jawed bony fishes, we get a spherical eye with a well-formed lens, and we get a crisp image. So that's showing the details of how the vertebrate eye developed. But again, the main focus of this particular slide is the fact that we've identified the gene that's responsible for the formation of crystalline that gives rise to the lens. And that has happened independently several times. So now let's switch to the topic of, it's a brief topic here in our chapter, of constraining evolution. That is, there are some adaptations that we might conceive of, we might think of, but they haven't evolved. They don't exist. It seems reasonable to think that they might, but they don't. So why haven't certain adaptations evolved? So for example, why are there no hawk-sized, dragonflies flying around? Why are there no 9-toed tetrapods? Tetrapods all have a maximum of 5 toes, and then in some lineages that's been reduced down to a fewer number. But why not? Well, physics imposes some limits. So that's the first point here. So we can't stretch the laws of physics. We're bound by the laws of physics. So in terms of the hawk-sized dragonfly question, well, that probably has something to do with oxygen levels. So back in the, insects breathe through the system of tubes, Malpighian tubules in their bodies. And so they can expand and contract a little bit, but it involves the diffusion of oxygen, CO2, in and out of these tubes. So yeah, so that's one thing, is a physical limit in terms of how oxygen can be delivered to the body. But history is interesting to look at here, because oxygen levels in the Carboniferous period, going back 300 and some odd million years ago, rose to about 35%. Today, if I'm remembering correctly, oxygen levels are about 20%, so significantly higher in the Carboniferous. And back in the Carboniferous, many insects were much larger. In fact, there were seagull-sized dragonflies flying around. So this would seem to support the hypothesis that the reason why, in some cases, we don't see some adaptations, for example, giant hawk-sized insects, is simply because it's physically impossible. But also here we're coming back to this concept of antagonistic pleiotropy. So mutations with beneficial effects on one trait those same mutations might cause detrimental effects on other traits. And so because of this antagonistic pleiotropy, that could impose limits on what is possible as an adaptation. Let's take as a point of example mammal cervical vertebrae. So that's what's being illustrated here on the slide and from your book, and an example of how antagonistic pleiotropy may be the reason why mammals almost all mammals have 7 cervical vertebrae in the neck. So sloths and manatees, as a matter of fact, have more than 7, a different number than 7. And even in giraffes, look at the figure, even with those long, long necks, they still only have 7 cervical vertebrae. They don't have more. And in fact, children, human beings born with an abnormal number of cervical vertebrae, are stillborn, are born dead, or they develop pediatric cancers and die early in life. Sloths and manatees, as it turns out, of course, have very low metabolisms. And very low metabolism, interestingly enough, is linked to low cancer rates. So there might be some antagonistic pleiotropy going on here, that with higher metabolisms, which is what us mammals have, very high metabolisms, with the exception here of sloths and manatees, slow metabolism. High metabolism, which is adaptive. We can be active all the time. But with antagonistic pleiotropy going on here, there's something happening with an increase or a change in the number of cervical vertebrae that genetically, with antagonistic pleiotropy going on, that gives rise to such developmental abnormalities such as pediatric cancers. So antagonistic pleiotropy may be the hypothesis to go for in some cases for why there is constraining evolution, why certain adaptations just don't exist. So here's yet another concept for us to chew on a little bit in this chapter: imperfections in complex adaptations. So because adaptations evolve from pre-existing structures and genes, for that matter, even complex adaptations can have deep flaws. In other words, we can't start off from scratch. We don't start off from scratch. We use the raw materials of the genes and the structures that already existed and build on that. We've seen many, many examples of that as we've gone along here in the semester. And but this topic of imperfections in complex adaptations is an extension of this idea that Evolution builds on what already exists. So, for example, the optic nerve in our eyes, in all vertebrate eyes, it plunges through our retina in the back of our eye and creates a blind spot. It's not very significant, but it's still a blind spot. It's still a patch in the retina that is not perceiving an image. And that's an imperfection, certainly not perfect. So that's an example. But as another great example, let's take a look at the recurrent laryngeal nerve that exists in giraffe necks as an imperfection, as like, well, used the raw material that was already there, and we end up with this crazy adaptation that certainly doesn't seem like, if you were starting from scratch, the best way to go. So in fish, as you can see in this figure, we've got gill arches that are delivering blood to the gills, and we've got nerves that are passing in between these gill arches. So that's the condition in fish, in vertebrates. That's how we started off. And so when tetrapods evolved, and tetrapods evolved a neck and lost the gills, well, these structures were already present. You can't get rid of them. You can't start from scratch. So the nerves and the blood vessels in tetrapods and a tetrapod neck were already present in fish. So the vagus nerve, which you can see in the figure, is found in both the shark and in us, or a giraffe, runs from the brain posterior to near the heart. And then a fourth branch of that nerve, which is now the recurrent laryngeal nerve, wraps around a blood vessel in the neck, or in the case of the shark, you can see it wraps around a blood vessel, and it goes up the neck to the larynx, to the voice box. The laryngeal nerve goes to the larynx. So check out that giraffe. Can't start from scratch. Got to use the variation that already exists. So you stretch that neck out, the neck gets longer and longer and longer, but the basic genetics and structural apparatus can't be changed. It's a constraining evolution there. And so we end up getting virtually a 20-foot long nerve that runs up the neck. It could be just a meter long, 3 feet long, if it didn't have to go all the way down and wrap around the vessels near the heart and then go back up the neck. So the overall bottom line message at the bottom of the slide, deep homology of this inherited developmental mechanism, meaning it goes all the way back to fish. That deep homology constrains the nervous and circulatory system anatomy, and there's no way around it. And that's what leads to imperfections in complex adaptations. So we're going to end our chapter here on from genes to traits on this topic of convergent evolution, because it pertains in this chapter on this topic. You know what convergent evolution is, when similar traits evolve independently in different lineages. But let's dissect this a little more carefully now that we have this background and understanding that in some cases the genetics for certain traits goes much farther back in history than the traits themselves. So mammals, marsupial and placental mammals, diverged from a common ancestor about 130 million years ago, long time ago. And both lineages, placentals and marsupials, eventually produce species that look remarkably similar and occupy basically the same niches. So by definition, that's convergent evolution. So we look at the figure on the left-hand side of the slide, in the both the placental and in the marsupial world, we have burrowers we call moles. in both the placental and the marsupial world, we have anteaters. An actual placental mammal we call an anteater, and then one called a numbat. And you can look at the figure, other examples, a mouse niche, a climber niche, arboreal, a glider niche, a wolf-like niche, etc. And then, of course, also even more dramatically, we've got wings in birds and wings in insects, convergent. Now, let's look at this in a much finer scale. I know we haven't done this yet in this class, and I know at least in Bio 182, most of us don't talk about parallel evolution. So sometimes in multiple populations of even the same species, multiple populations independently evolve the same trait in parallel, shifting independently. They're independently doing this from one another, from the same ancestral trait to the same derived trait. So that's called parallel evolution, when temporally, time-wise, it's happening from an immediate common ancestor basically at the same time, whereas we would use the term convergent evolution for the evolution of similar traits in very different ancestors, far away from each other in a phylogeny. So a good example of parallel evolution, and there are many of them, are cave populations of astaninids, fish. It's a tough one to pronounce. And in fact, do I still have it here on my iMac? Let's see.
Astyanax.
Astyanax, there you go, pronunciation. Astyanax. In Mexico, independently evolved pale body color and reduced eyes, living in caves. So they're part of the same species, but they have these populations of independently invaded cave systems. And through that independent invasion of cave systems, those populations have evolved again parallel, pale body color and reduced eyes. So this kind of brings us to a good question. When is a trait convergent and when is it parallel? I think the distinction can more or less be made by identifying what the common ancestor, how close the common ancestor was for these lineages that evolved the same trait. But still, it becomes a little bit dicey, you know? It's not a clear-cut objective thing. So, for example, eyes evolved independently in arthropods and invertebrates. We have very different kinds of eyes. But, as we've seen in this chapter, the underlying genetic mechanisms are the same genetics. They didn't evolve different genetics for similar traits. It's the same genetics. So some biologists, Cliff Tabin and Neil Shubin and Sean Carroll, have come up with the term deep homology. Deep homology, referring to these cases, these instances, where even though the traits have evolved independently in very distant lineages, the genetic mechanisms for those traits is homologous, is the same. So they refer to that as deep homology. So the evolution of our limbs in insects and in vertebrates, deep homology. The evolution of eyes, deep homology. Useful concept for understanding convergent and parallel evolution. So there you go. This ends our chapter on From Genes to Traits, and you can see we've traveled through many different topics and concepts here. And in some cases, we've come back to concepts that we've visited before, like convergent evolution, and adding some new spins and some new understandings to these topics as we build our understanding of evolution and the basis for how evolution happens, particularly, like we've been doing quite a bit, the genetic basis of evolution.